The overall goal of this procedure is to generate myospheres, EB-like structures composed of skeletal muscle cells, from human embryonic stem cells. This is accomplished by reprogramming the cells by lentiviral infection of BAF60C. The second step of the procedure is to infect the same cells with a second virus, the MyoD lentivirus. The infected cells are then induced to form EB-like aggregates for 5 days in serum-based culture medium. Then, the EB medium is exchanged for a serum-free differentiation medium, and the aggregates are grown for 2 more weeks into myospheres. Ultimately, immunofluorescence is used to visualize sections of OCT-embedded aggregates.
The main advantage of this technique is the high yield regeneration of skeletal muscle cells that, unlike derivation from mesenchymal or mesodermal progenitors, does not require FACS sorting and is therefore compatible with a three-dimensional culture system.
The day before the infection, to human embryonic stem cells in 6-well plates, add human embryonic stem cell cloning and recovery supplement to the medium at 1000 x for a final concentration of 2 micromolar. The next day, infect the human embryonic stem cells with high-titer BAF60C GFP lentivirus. First, dissociate a well of cells into a single-cell suspension by adding 1 milliliter of TRIP-LE